I'm Dr. Andrew Favich, and it is my honor to be your host for this presentation this evening. Logos Research Associates is a fellowship of scholars and scientists that are committed to upholding a high view of science and a high view of Scripture. We sincerely believe that scholarship, logic, and the scientific method can and should be used to show that the Bible's historical claims are both credible and ultimately superior to the secular model of origins. Logos sponsors many creation research initiatives and projects involving many different experts and scientific disciplines, all working towards the goal of increasing the explanatory power of the creation model of origins and of Earth history. This goal of this live stream series is to provide you with the opportunity to hear from top researchers who will convey cutting-edge scientific evidence for the creation account. Before I introduce our speaker to begin our presentation, we will open in a short prayer. If you would, please join me. Creator God, thank you for this wonderful creation you've given to us to explore, and the depths with which we can explore it are so vast and amazing that even at a level as small as the molecular level, we can even see your fingerprints all over this creation. Father, I ask that you would open our minds and hearts as we look at this world you created to stand in awe of what you have done and walk away closer to you. We ask it all in Jesus' name. Amen. So I now have the pleasure of introducing our speaker. Dr. Joe Deweese earned his BS in biochemistry from Freed-Hardeman University in 2004 and his PhD in biochemistry from Vanderbilt University in 2009. He taught pharmacy school at Lipscomb University College of Pharmacy for 12 years in several content areas, including biochemistry, physiology, pharmacology, ethics and values, and laboratory methods. In August of 2021, he and his family moved to Henderson, Tennessee to teach at FHU. He's professor of biochemistry and director of undergraduate research at FHU. Welcome to the broadcast, Dr. Deweese.
Thank you, Dr. Favich. It's good to see you, Andrew, and appreciate you being here with us tonight and hosting. So thank you very much.
Likewise.
Well, I'm looking forward to it. And I'd like to share with you all some things, some thoughts that I have about science and research, but particularly looking at molecular sciences. So definitely looking forward to this. So thank you all for being out there. I definitely prefer a live audience. And so I hate that I can't see you, but I'm going to pretend that there's a couple of you out there listening and hopefully learning something from what we talk about tonight. So I want to talk to you about kind of what I consider design in the unseen world of the cell. And I'll explain what I mean by that as we go along tonight. But let's first start with some of the terms and things that we're using, like this term molecular. What do we mean by molecular? Maybe in your mind you picture, I don't know, what do you picture? What does a molecule look like? And maybe some of you have used molecules like what's on the screen here in a class, chemistry class or something like that, where you're able to actually pull them apart and look at the individual molecules. And the reality is these molecular toys that we have here on the screen are ultimately made up of millions of atoms themselves. And so these atoms all bonded together in particular patterns. And so I'd like for us to think about what is life like, though, at that smallest level, at that simplest level. And really, we think of it in levels of organization. The organism, of course, humans or animals, other creatures, is organized into different groupings. For instance, organ systems. We have a number of organ systems in our body that contain organs or organized with various organs that carry out particular functions. So maybe the cardiovascular system or the muscular system, digestive system, and so forth. But within these various organ systems, we have tissues which make up those organs. And that's the actual groups of cells that in layers of tissues that make up, let's say, the liver. And so as you dig into that and you look deeper into the liver, you find that these tissues are made up of individual cells. And then the cells have compartments within them that contain various molecules that are carrying out the various functions of life. And so in our little schematic here, we have DNA in the nucleus. And then, of course, DNA itself is composed of atoms. Now, we have a very generic drawing of an atom here. I know that's not what they would look like if we were able to zoom in on them, but the concept there is the idea that at the fundamental level, atoms make up molecules, and these molecules come together to make up the compartments of our cells. Cells come together to make up tissues. Tissues come together to make up various organs, and then combine together into organ systems, and all together into the organism. And so humans have on the order of trillions of cells in our bodies that make up these different tissues and organ systems. And I want to talk to you tonight kind of down at that cellular and really the subcellular level about what's going on inside those cells. If we think of it in terms of scale, if you think of a meter, just over 3 feet, we can kind of get a sense of how small this is. Most of us probably by visualizing or looking at a ruler can conceive of things like a millimeter, right, as a fraction of a meter. And really that's small, but that's not super small, right? We can go below that, right? You can go to what we call micrometers or micrometers, and that's a millionth of a meter. And then a nanometer, right? That's 10 to the minus ninth, and even a picometer. So you can keep going. And we're not going to go deeper than that right now because this really conveys the area where we're at. Human cells are in the micrometer range. And when we get down to the protein level, we're somewhere down near the low nanometer range. And atoms themselves are just below the nanometer range, kind of in that range of between picometers and nanometers. So we're talking about very, very small things that are below the ability of kind of visible wavelengths of light for us to even see these things. And so it's really kind of an interesting concept to consider how small these things are that we're talking about. But I wanted you to kind of have that in your mind, and we'll come back to that. So file this away. We're going to come back to this just a little bit later. As we think about the molecules of life, living cells are really made up of kind of 4 groupings or categories of molecules. And I put some representative examples here on this slide for you to consider. For instance, proteins are made from amino acids, and there's a couple amino acids there. We also have carbohydrates or sugars are a major part of our structures in our body, both from an energy perspective, but also from the perspective of just being able to signal and communicate information and so forth. We also have lipids, fats, that are used to store energy, and they can be burned up to release energy. They also make up much of our membranes. Cholesterol there is shown as well, which is a major component of our membranes. I know we've demonized it, but we really got to have it. We have to have a certain amount of cholesterol. In fact, our cells will make more if we need it. And then you have things like nucleotides, the building blocks of DNA and RNA molecules. And this one is an example of a ribonucleotide. So these would be kind of the 4 fundamental classes of molecules. And then there would be many more within each of these groupings that would be derivatives and various versions of molecules like this that make up all of the parts of our cells. Now, tonight we're going to focus mainly on those amino acids in the form of proteins, because we're going to be talking about enzymes and molecular machines, the vast majority of which are made of protein in our cells. So we'll talk about and kind of build upon that as we go tonight. So I want to have this thought question for just a moment of what do we think about when we think about a living system like a cell, an organism, so forth? What are the requirements? What is needed for there to be a living system altogether? And I think one of the things that we come to when we think about this, there's lots of ways we could go with this, but where I want to direct your thought to for the moment is this idea that living systems, on some level, we've got to figure out what is the smallest fundamental unit that we kind of consider to be alive. This debate obviously rose up again in discussions in the last few years with SARS-CoV-2 and that virus. Our virus is alive, right? We anthropomorphize them in our language very commonly. But I think most of us would agree in general when we look at that a virus on a fundamental level is not alive in the way that a cell is alive. And so where is that dividing line, and where do we kind of divide those things up is an interesting question to consider. But I would like to establish, or at least let me take for granted this idea that cells are the basic unit of life. These are the fundamental units that are living, growing, dividing within us. We could use lots of different analogies about cells and about their functions. They're much more than simply machines, and they're very different than any machines that we create, in the sense that these things can actually essentially construct copies of themselves and duplicate themselves. And it's really a fascinating thing to think about what cells are capable of doing. In fact, this is from a biochemistry textbook. This happens to be the one I had in college, from Lininger's third edition here. A living cell is a self-contained, self-assembling, self-adjusting, self-perpetuating, constant temperature system of molecules that extracts free energy and raw materials from its environment. Let that sink in for a moment. Of course, this textbook does not come from a design perspective, a creation perspective. They're just making an observation here about cells. They're self-contained, meaning in the sense that there is a compartment here that has a boundary or a barrier. They're self-assembling, so there is somewhere information governing the construction of various molecules and the arrangement of these molecules in relation to one another. It's self-adjusting in the sense that it responds to what's going on inside and outside of the cell. Self-perpetuating, meaning what? It can actually make copies of itself or replicate itself and divide into two new cells. And a constant temperature system. So there is a process by which cells are trying to regulate what's going on within them in relation to their environment. And so it's really a fascinating thing. They extract free energy and raw materials from the environment, meaning cells are actually drawing molecules in, using those materials to make energy, removing waste products, sending those back out of the cell. All of this is taking place in the cellular system. And we could describe them in human terms as, a factory, a trash compactor, a recycling system, a manufacturing system. All of these things would be appropriate, but really pale in comparison to what's actually going on in a cell. There's a lot of different things happening here, a lot of complexity going on within the structure that we call a cell, that here is kind of, diagrammed in a very cartoony way. But the concept is that within this cell, you have not only the genetic information contained in the nucleus, but you have all the various components needed to carry out the functions of the cell, and needed to make energy, make more molecules, make more proteins and enzymes, etc. All of that is contained within this system. And that's pretty fascinating. If you really think of it on another level, this cell that contains, let's say, the human genome in it, not only can become this cell, but it has the information and the capacity to become any cell type in a given organism, right? So if it's a human cell, it could, in theory, become any cell type. Now, obviously, that's a little harder to do in a practical application of taking one cell and converting it to something else. That gets a little more complicated. But the concept is the information content is there. And these cells can do these types of things, which is pretty fascinating. I want to focus in on one of the components in our cells, and that is the proteins that we refer to as enzymes. Now, maybe some of you, when you think of protein, you think of the nutrition facts on the side of a box of your favorite Pop-Tarts or cereal or whatever. And you think, just protein is a generic, as a concept. But I want you to realize when we talk about living systems and cells, and we refer to proteins, we're oftentimes referring to very specific versions of these molecules and what they do. They have very particular three-dimensional shapes and structures, as I'll show you some tonight. And they carry out very specific functions. Some of them are catalytic, meaning they actually can break and make bonds. And those are the ones that we refer to as enzymes. Other proteins might be more structural in nature, where they are perhaps connecting things together and building larger structures. And so I want to focus in, though, tonight on this idea of enzymes and the types of things that enzymes can do. So I want you to think of enzymes as proteins that can build up, break down, or interconvert molecules. So if I go back to our little molecular kit analogy earlier, and the idea that we could pull any of these molecules apart at any given time and separate them apart, I want you to think of that as that's what these enzymes are able to do. They're able to pull molecules apart and put them back together, or put them together in different arrangements, or add things to them that weren't there to begin with. These are the types of things as we think about enzymes, as we think about the functions that they're doing. That's kind of what I want you to picture in your mind. And then as we kind of zoom out for a moment, this is taking place all throughout our cells, making all kinds of different products and things like this. Now, this is a kind of a broad biochemical pathway that includes some things that maybe humans aren't per se metabolizing and making, but these are molecules that are made in various living systems by means of enzymes. And what's cool is something I'm not going to get to tonight is that a lot of these pathways are interconnected. It's not just a random enzyme picking up a random substrate, converting it to a random product. There actually is a lot of intentionality and purpose behind enzymes and the pathways that they work within and the types of molecules that they create. It's not completely haphazard. It's actually quite intentional. And so we'll perhaps talk about that another time. But I want you to consider that enzymes are required for really essentially every fundamental process that we think about cells being able to do, whether it's copying the DNA, making RNA from DNA, we call that transcription, making proteins from RNA sequences, we call that translation, breaking down molecules, building up molecules like sugars, lipids, proteins, etc. All of these processes require enzymes. Making energy to carry out all of those processes above requires enzymes. Communication for cells to make a particular, let's say, transmitter, neurotransmitter, and send that molecule to other cells to convey a particular message. That oftentimes requires multiple enzymes to carry that out. Enzymes are involved in transportation, import, export, getting things into and out of cells and compartments within cells oftentimes requires enzymes. And we could go on. So I just want to, I don't want to belabor the point, but I just want to make the point that enzymes are really critical. They're a huge part of what takes place in cells at any given time, and they're required for cells to function. So if we don't have enzymes, we're not alive. This deal doesn't work, because these things are absolutely critical to what goes on within a cell. So let's just take, for instance, I don't know who all went out to eat tonight, maybe had dinner, maybe had a burger, and perhaps you don't think about what happens to that burger as your body digests it. This is one of the nerdy things that some biochemists do. I'm not going to name any, but I will say some of us think about what is happening to this food as it goes through your body. Now, maybe that grosses you out. Okay, so we're not going to take this too far, trust me. But I do want us to think for a moment about when we start eating, our mouth, our digestive tract starts to break that food down. And we have various enzymes that are breaking the molecules of the food up. Yes, I know we chew in our mouth, but that's not the end of it. That's kind of the beginning of the process of breaking this down so that we can extract from it whatever molecules we might be able to use. And so we break down what we might call macromolecules. So maybe that's long chains of sugars, maybe that's proteins, maybe that's various lipid structures. We're breaking some of these things down. And then we're actually going to, as it passes through our body, we're going to start absorbing these things. For instance, in the intestines, we're going to absorb these molecules really as kind of their monomeric components, meaning the individual components of some of these structures, like the individual sugar molecules, amino acids, and so forth. They're getting absorbed by our gut, by our stomach. Many of them get repackaged in that context and then released into the bloodstream where they can circulate. For instance, this is going to cause a rise in blood sugar levels. That's going to be detected by the pancreas. The pancreas is going to respond by decreasing production of glucagon, increasing production of insulin. And the result of that then is ultimately that we've just started to really take care of what's going on in our body to respond to it, so to speak. Perhaps some of you know people, or maybe some of you deal with diabetes. Insulin is produced by the pancreas in response to a rise in blood sugar levels, and it regulates not only how our body metabolizes sugar, but also how we metabolize lipids as well. And so a dysregulation of that pathway leads to multiple problems. Glucagon is kind of the competing hormone. So hours after a meal, your insulin levels will go down, your glucagon levels will go up, and your body will respond by releasing some of the stored energy that has been put away for later from the meal that you ate. So there's a lot of things going on. But what if we just kind of zoom in for a moment and let's just look at that example of sugar for a second and ask, how does the sugar from this burger end up in any way benefiting us, right? Because, I mean, this thing looks like it's way too good to be healthy. So is there any redeemable quality in this burger, right? Now, I would say there is, okay? I don't want to take it too far. But as our body breaks down the components that are in this food, let's just say, for instance, that some of that is carbohydrate, and it is, obviously, and we're going to get some sugar out of that. How does our body respond? We already mentioned insulin. Here's a ribbon diagram of the structure of insulin. Now, for comparison's sake, insulin and glucose are not the same size, okay? So I show them here looking about the same size. I want you to think of the glucose molecule as smaller than the arrow on the aqua structure there of insulin. Okay, so it's going to be small, fairly small in comparison to the overall structure of insulin. Well, insulin signaling is going to cause a response throughout your body. So as that insulin released by the pancreas is going to be distributed by the bloodstream, we call that an endocrine hormone. And endocrine signaling is going to go throughout the body. And so insulin is going to bind to receptors on the cells throughout your body. Interestingly enough, they're called insulin receptors. How about that? Some things we do make simple. These insulin receptors then are going to elicit a response within those cells, whereby those cells are going to try to get this sugar into the cells. And so, for instance, we're going to take glucose transporters, which have been sequestered, and put them out at the surface of cells so that the glucose can be imported into those cells. Once glucose is imported in, we carry out processes like glycolysis, and then we take that glucose that we've broken down to pyruvate during glycolysis. We convert it from pyruvate to an acetyl-CoA group. We take that acetyl-CoA and we send it into the mitochondria. Actually, it's in the mitochondria already. And we send it through the citric acid cycle. We harness a bunch of electrons from that, and those electrons pass through various electron transport complexes in the membrane of our mitochondria, the inner membrane of our mitochondria. And then they create a proton gradient that powers a pump that we call the ATP synthase, and voila, we convert ADP to ATP. So I know this is backwards here, so just imagine that going the other direction of my little schematic here. So we make ATP as a result of that. And so this whole process is carried out thanks to dozens of enzymes. Okay, the glycolytic pathway has 10 different steps, 10 different enzymes involved in those steps. You've got pyruvate is the pyruvate dehydrogenase complex, which converts pyruvate to acetyl-CoA, is a multi-enzyme complex that carries out that function. The citric acid cycle is 8 steps and 8 different enzymes carrying out the various steps of that. Over into the electron transport chain, you have multiple protein structures, protein complexes, passing electrons through and protons through very carefully in a very ordered manner so that you can ultimately power that enzyme there at the bottom, the ATP synthase, and generate ATP. Okay, so question. All those pathways are just there because of random mutation and natural selection, right? I mean, that's it. That's all it takes to give us each of those well-orchestrated pathways. After all, they are present throughout all the domains of life. in some form. And so clearly, natural selection can just do that, right? I know, and I'm guessing there's probably some engineers listening out there. And if we offered such a haphazard, random, undirected explanation as to how this phone was constructed, we would be laughed out of the room, right? We would be laughed off the internet, because there's really no way to go from the raw materials that make up this phone to get all of the integrated circuitry and the battery and the screen with all of its abilities. That just doesn't happen. And I think on a fundamental level, we all understand that doesn't happen for phones, right? This is Paley's watchmaker argument. This doesn't just happen. Why is it that when we look at the living system, we just assume, well, that must just happen? I would argue that the living system and these components that we're talking about are very complex, and more complex than this phone that we're looking at. All due respect to the engineers that brought this into being, we are far more complex than this by orders of magnitude. Take this little pathway over here, glycolysis. This isn't the greatest illustration. It's just one I could find that was available to show for free. But you've got a series of 10 steps. Okay, we're going to start over here with glucose. We're going to phosphorylate it. Becomes glucose 6-phosphate, and then we're going to convert it to fructose, and then we're going to cut it in half, or phosphorylate it again, and then cut it in half. And then those two molecules become one particular version of that, which gets phosphorylated again and goes through a series of steps, giving us at the end over here pyruvate. And what you notice is some steps cost energy. A couple of these early steps cost energy. A couple of the steps at the end actually make energy. And we actually end up with 2 molecules of pyruvate, because if you're paying attention here, there's actually 6 carbons that come about from this event where we've cut this thing in half. And those come down here and kind of close out this pathway 2 times. So each of these steps are really, really critical. Some of these thermodynamically are pretty reversible, meaning the enzymes could actually go back and forth. But there are several steps in this pathway, 3 steps in particular, that are heavily regulated to control the flow in the progression of this. I will show you here one example of an enzyme in this pathway. There's so many we could show. We could spend our whole time there. But glucokinase is an example of an enzyme that is part of this pathway at the beginning there, where you actually take the glucose, and when it's brought into the cell, glucokinase will add what we call a phosphate group to it. So the enzyme is the colorful ribbon diagram that we're looking at here. The sugar molecule you can see here is quite small by comparison, right? This is glucose here in the middle. And then this little structure right here is ATP, where we're actually going to take a phosphate group off the end of that, thanks to a metal ion that we've got in here helping out. And we're going to put that phosphate group onto glucose. And it requires all of this structure to do that. Okay, this is a fairly simple reaction by comparison. But yet we have to have all these different features to do this. And what's cool is there are different versions of what we call hexokinases in our bodies that are present in different tissue types and have different levels of affinity, so that each cell gets the right amount of glucose and not too much, which is really fascinating. So it's not that there's one glucokinase to rule them all, right? Or one hexokinase to rule them all. We have multiple versions. And they appear to be fine-tuned for specific tissue types, which is really fascinating. But let's look at this next step here, the pyruvate dehydrogenase step, where we go from the pyruvate to acetyl-CoA. That's carried out by this enzyme that we call the pyruvate dehydrogenase complex, which is quite fascinating. This complex is a multi-enzyme complex that has 3 different enzymes, but multiple copies of each of those enzymes. And this particular image comes from the Protein Data Bank. It happens that this was the molecule of the month several years ago. And they even state in their description of this particular structure that this is a simplified version. Only 6 of the 24 tails on this structure are shown. The center of the core is actually completely surrounded by these structures. And I don't have time to go into the details of the actual mechanism of the reaction, but it's really, really fascinating what happens in this enzyme and how it carries out what it does and then resets to do it again and is able to do that over and over. So that's one of the cool hallmarks of enzymes, is that they're able to be used again and again. And that's really well illustrated in the example of pyruvate dehydrogenase. But let's look at one other component of this for just a moment, and that's the ATP synthase. This is the actual motor here that's making ATP. So there's a portion down here at the bottom in this depiction where protons are able to flow from what we call the intermembrane space into the mitochondrial matrix. And as they flow through this rotor here, they cause this thing to spin, which spins this blue portion in the middle of this cap structure. As the blue portion spins, the cap structure is able to convert ATP or ADP rather to ATP. It's really fascinating. Let me show this little animation where you can kind of see that, where you can see that kind of happening in an animated way. So you can see the protons are concentrated here at the top. The protons are flowing in and around this rotor, and that's turning this central shaft inside the cap domain. So this is kind of upside down relative to the last image I showed you. So as these protons flow in, they go around the merry-go-round of the rotor, and then they're able to be released on the other side over here in the matrix side of the membrane. So the matrix is kind of the very innermost part of the mitochondria. And as that happens, that spins this little shaft, which then can basically, deflects these little heads here, for lack of a better way to describe it. As those conformations change, those shapes change, they're able to take ADP and a phosphate and convert it to ATP and then release it. And so there's a symmetry here where there's actually 6 subunits here, and this reaction is going on 3 times as it goes around here. So there's a active site here, one here, and one on the far side. And so they're going, as that thing goes around, it's able to convert the ADP to ATP. This full animation is available on YouTube from Discovery Institute, so I recommend you check that out. And you can get the full description and details of what's going on with this little motor. It's really, really fascinating how this machine works. And again, there's many of these all together in our mitochondria, operating all the time, allowing those protons to flow through that little rotor there to continually generate ATP. So, question: what forces could bring about such interrelationships? I think it's a fair question, and I think it's one that has to be addressed. Where does this type of structure, these types of systems, these types of enzymes, where do they come from? They work together. They're highly orchestrated. The pathway I just described for you, from glucose import through glycolysis into the mitochondria, pyruvate dehydrogenase complex, onto the citric acid cycle, to the electron transport chain, to the ATP synthase, excuse me, again, requires dozens of enzymes. Where did all these enzymes come from, right? How did they all come to work together in such a highly orchestrated manner with a very goal-directed purpose in mind? This is not haphazard, right? This is very intentional, very directive. And I think we need to recognize and acknowledge that. All right, let me shift gears for a moment here and go into a realm that I spend most of my time in, and that is studying DNA and enzymes that work on DNA. So I don't study metabolism. I teach it a lot, and I love the application and illustration of it, because everybody relates to eating and being hungry and hopefully enjoying food. And so I think there's a relatable nature of that. But I want to look at another area where enzymes play really critical roles for the remainder of our time. This is DNA, just a short segment of it. Okay, this is just a small snippet of it. And I put it here just for a moment, just for us to look at and to think about this structure. So you notice a couple things about the structure of DNA. First of all, it's two strands. So as it rotates here, I'm going to play this again. I want you to see there's two strands, and they're actually running in opposite directions. It's a little hard to tell, just by looking at an animation on the screen, but they're actually running in opposite directions, and they wrap around one another, forming what we call the double helix of DNA. Okay, so there's two strands wrapping around each other. All right, on the outside, we have those phosphate groups. And then between the phosphate groups, which are kind of the orange and red, between the phosphate groups, we have the green, pentagon-shaped structures here, which represent sugar. That's, a ribose sugar. And then in the center, flat relative to the view that we're looking at here, are the actual DNA bases. And so perhaps you've heard of A, T, C, and G. Those represent the actual bases of what we call DNA. And so it's the arrangement of those particular bases in a given order that give us a sequence of DNA. Now, how long are sequences of DNA? They can be huge. Okay, our genome, we have 2 copies of it. So it's a 3 billion base pair genome times 2. So we have 6 billion letters in our cells that are arranged like this. Obviously very, very small because we could fit them all in there. And this right here has maybe 10 base pairs, and we're talking about billions of base pairs in our cells as a whole. In fact, if you took all the DNA out of your cells and just take one cell, for instance, and take all the DNA out and stretch it out end to end, it would be 6 feet worth of DNA, about 2 meters worth of DNA. It's really fascinating to think about how much information is crammed into a cell, which is, as we talked about earlier, on the order of a millionth of a meter in terms of scale. So it's in the nanometer range, the micrometer range of scale. So really, really tiny structures in our bodies. It's absolutely fascinating. One other thing I'll note here real quick before we come off of the structure is that what you'll see is that the bases are pointed to the middle, and there's some differences you can see. We have what we call the major groove, which would be this section, and then the minor groove, which would be this back section. So we call them the major and minor grooves of DNA. So here's the major groove along this axis right here, and then the minor groove along this back axis back here. And that's important because when we think about how proteins interact with DNA, many of them have to bind along this major groove section to actually be able to recognize the bases. Because if they just bind out here, it's all phosphate groups. They actually have to get into the groove here to actually recognize a particular sequence of DNA. There are things that can bind in the minor groove, but I think for the majority of proteins that interact with DNA and are site-specific, they need that major groove interaction to really get in there and see what's going on inside that. So, hope that's helpful. I just thought that'd be a good way to kind of illustrate DNA and its structure. Now, let me offer to you though, DNA doesn't just exist in these double helices. It actually gets organized and compacted, which is why it fits in your cells, as we were talking about earlier. So the DNA actually wraps around proteins that we call histones and makes these coils. It actually does 2 turns around this, and it's roughly 140 to 160 base pairs to get around this particular structure. There's actually 8 proteins in the middle here. You can't really differentiate them, but that would be the core of a histone octamer. And then multiple of those come together. So you can see those. We call those kind of beads on a string structure there. And then that wraps up tighter into what we call a fiber. And you can see all the little beads here wrapped up tightly. And there it is in terms of structure there. And then there are scaffold proteins that allow that fiber to fold into particular shapes. And then those condense further with additional proteins until you eventually get to the chromosome structure that maybe you're familiar with seeing in the form of a chromosome spread. Sometimes we see chromosome spreads where you can actually see all of the individual chromosomes of a particular cell. That's when they're in this state. Now, this is a highly compacted state. So this is kind of like that information is closed off. It's not really available for use per se at the moment. It's really just kind of basically packed up for moving. All right. So this is kind of the cells moving truck version where we've got everything packed up. But during the rest of the cell cycle after cell division, certain portions of that are going to become decompressed and opened up and accessible for utilization by the cell. So there's a lot of compaction that goes on, but it has to also be reversible, where you have to be able ultimately to get back to the isolated DNA regions themselves and use that information. So I want us to have that kind of in the back of our mind as well. Okay. All right. Now, another thing to think about is, all right, how does all this information work together? We think about it. We have what we call the central dogma, which is this idea that DNA can be transcribed to make RNA, and RNA gets translated to make protein, and then protein makes various metabolites and macromolecules. And we've coined terms in the last few decades to describe some of these things, like the genome is all the DNA, transcriptome would be all the RNA transcripts made in a particular cell, the proteome would be all the proteins made in that particular cell, and then the metabolome would be all the small molecule metabolites that a particular cell type might produce. And then, of course, replication would represent the copying of DNA. While this process is true, it is now better understood that there are a lot of other things going on here as well. Like RNA can actually influence the transcription and translation of various RNA molecules and proteins. We also know that proteins influence transcription and translation, and metabolites influence transcription and translation. So there's a lot of, in some cases, feedback could be positive or negative, but there's a lot of influence from these other components on the process as well. And then we do know that in some cases, reverse transcription also takes place where RNA can be made into DNA. So that kind of gives us a little concept that while DNA to RNA to protein still holds true in general, it's not the whole story. And there are other components going on here as well. Okay, let's look at for just a moment. So right here, this step of DNA being copied, replication, what does it take to copy DNA for a moment? I heard a fascinating presentation by Dr. Jonathan McClatchy a few weeks ago about DNA replication. And he argued, and I agree, that replication is essentially an irreducibly complex system. In this particular model here, they're showing you, for instance, the need for several enzymes, topoisomerases that we'll talk more about later, helicases, single-strand binding proteins, various polymerases, a primase to make an RNA primer, a ligase to attach fragments back together, another DNA polymerase on what we call the lagging strand. So you got multiple components that all have to be there. And this is true at every level of life. So even the simplest living organisms require these components in order to copy DNA. It's not just one protein that does it all. It's multiple enzymes, all coordinated, all working together. And there's more proteins that we're not even showing here that are involved as well. And I think I would argue that you could dig into this process and see that it is, in fact, irreducibly complex. There's a certain minimum number of components that you've got to have or the system doesn't work at all. And I think that's pretty fascinating to think about. I would argue that it's what we call irreducibly complex and interdependent. So irreducible complexity, as defined by Behe, he says, by irreducible complexity, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of these parts causes the system to effectively cease functioning. And I think you can make the case that if you start removing components from this, the system's going to fail, and you're not going to get DNA copied efficiently, and especially efficiently enough to sustain life. Now, interdependence is another concept I want to layer on top of that, and that's this idea that there's a causally circular relationship between biomolecules. In other words, the products of the process are required to carry out the process. Let me illustrate this just with one quick note here. If we want to copy DNA, we actually need about 30 different proteins for synthesis. There's really about 160 proteins involved in replication and repair in humans, but you got to have about 30 for synthesis. One of the simplest known living organisms, Mycoplasma genitalium, also requires about 30 proteins. Transcription from DNA to RNA, about 40 different proteins, at least 10 in Mycoplasma genitalium. Translation requires over 100 in Homo sapiens. Still, in Mycoplasma genitalium, to make a protein, you have to have at least 100 different types of proteins. Think about that for a second. I got to have 100 different types of proteins to make protein. That's causally circular. And even energy production, over 70 different proteins, those pathways we were talking about earlier. Mycoplasma genitalium needs at least 30. Now it skips some steps because it actually, as a kind of a parasite, it's feeding off a host here. So it's kind of cheating because it's getting some of its resources in a little different form than free living organisms, but still illustrates the point. You got to have a lot of proteins, a lot of things. So the question is, okay, if DNA is needed to make RNA, which is needed to make protein, but we need proteins to make DNA, RNA, and protein, which came first, right? This is a chicken or egg type concept that we have to deal with if we're going to understand where all of this came from. They all had to be there present, working together to make this system work. And I would argue that in biological systems, irreducible complexity and interdependence are the rule rather than the exception. This is what we see over and over again. And I think it's really important for us to consider that. So let me ask this question as we kind of come to the final kind of section here. How do we study what we cannot see? As I mentioned earlier, these things are really, really tiny, and light microscopy won't cut it. But there are some other forms of microscopy that can get us closer. But how do we study these things, these molecular machines that are down at this really, really tiny scale? I want to give you from Romans chapter 1 and verse 20. Paul's writing here to the church at Rome. It says, the invisible attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world in the things that have been made, so they are without excuse. I understand there's a greater context here, so I don't want to take this out of context. But one of the points that Paul makes here is that how do we know God created this, right? How do we know he was involved in this? He says we can see in the things that have been made God's divine attributes. In other words, we see evidence of his working, of his power through his creation. And I would like to suggest that one of the ways we see God is we see the effects of God. We see the things that he has done. And so I would argue in the same way that in the laboratory, we study things that we cannot see by measuring their effects, right? What effects do they have on the things that they're working with? So we use relatively simple methods oftentimes in order to observe these effects and actually measure and detect these. So I want to tell you the story briefly here as we close, because I want you to come back in June and hear the rest of the story here. But I want to tell you briefly part of the story of what I call the reversible molecular scissors. That is these enzymes we call topoisomerases. In particular, I like this one. This is my favorite, DNA topoisomerase 2. And the 2, we will explain a little bit more in the next talk why it's 2 and not 1. There is a topoisomerase 1, but I think topoisomerase 2 is way cooler, all due respect to topo 1. But this enzyme, I'm showing you here a couple different views of it, is made of 2 different proteins in humans and eukaryotes. It's made of 2 different proteins that come together. They're actually 2 copies of the same protein. Let me clarify that. 2 copies of the same protein that come together. Now I've depicted one here in kind of this gray color and the other one in a colored ribbon diagram. And then you can see a piece of DNA in green kind of through the middle here. And then I've turned them a little bit so you can see it. So topoisomerases are these cool enzymes that alter DNA topology. So they don't change the components, but they do change the structural relationship. They isomerize molecules, and the ACE on the end is an enzyme. So it's a topology altering enzyme for DNA. And what do we mean by DNA topology, right? DNA topology has to do with knots and tangles in our DNA. And everybody's familiar with knots and ropes. In fact, we all wish that if we had a shoelace like my little string here, that was knotted, right? We all wish that we could just remove that knot and just be gone with it. And one of the cool things is that we have in our cells a machine that ultimately can do that for our cells and just remove that knot right out of it. And that's what these topoisomerases do. Now, why do we need this? Why is this a problem? Remember, we said we've got about 6 feet worth of DNA in each cell, and our DNA is double-stranded. And anytime you try to open that to access it, just take a double-stranded rope and try to pull it apart. You're going to get all kinds of coiling that's going to result in what we call supercoils. Now, those of you who perhaps are of a generation that had phones on the wall that had a cord coming down, you remember the one in the kitchen, right? That every time you went to answer it and you pulled it off the receiver, your head went straight to the wall because the receiver was so wound up in the coil of the telephone cord, you couldn't even pull it away from the wall. And that's a supercoil, and that's happening in our cells to our DNA. It's really fascinating. And so we have to alleviate these types of things in order for our cells to survive. And so, for instance, we can actually monitor this in experiments. So DNA that is supercoiled, it's all twisted up, runs faster in this particular type of gel that I'm showing on the screen than a piece of DNA that is what we call relaxed. Now, these represent what we call plasmids, which are DNAs that are connected on the ends. Our chromosomes are not, but in this example, these are. And then the intermediate components are what we would call the topoisomers, where they're in between this really, really wound up state and the fully relaxed state. We can actually see a difference. And so we can actually experimentally measure changes in that when we change things about the enzyme. So here's a mutant that really didn't see much of a change. Here's one where we see one enzyme actually seems to work better or faster than the wild type. This one seems to work slower. And so we can study these mutations based upon these effects that we're seeing in our gels and try to understand what's going on and why are these things maybe working faster or slower. This work was something we published a few years ago. We're still following up on some of these. And so it's been a really exciting thing to study, and I'll share more of that story with you in the next talk. But topoisomerases are really fascinating. So let me kind of walk you through how they do what they do, and I'll show you a quick animation as we get ready to close. So topoisomerases, again, are these two subunits. They're able to bind to one section of DNA here in maroon, and then bind to a second section above that. They break the maroon section in half, pass the gold section through, put the maroon section back together, then release the gold, and then release the maroon. And so essentially, if you have two things that are interwound and kind of wound around one another, they can undo that, alleviate that through this mechanism. So by this mechanism, topoisomerases can relax DNA, they can unknot knots, they can unlink links between chromosomes. The type 1 topoisomerases can also do relaxation like the type 2 topoisomerases. But the type 2 topoisomerases have an advantage in that they can also unknot and decatenate DNA, which is really cool to see. So let me show you this quick animation, kind of show you the concept of what these things do. So here we have our enzyme coming in. You're going to see two segments of DNA come into the screen here, one binding in the active site, the second binding above that. We're going to make the upper one transparent for a moment, so you can see the break in the lower one. Now we're going to pass the upper segment of DNA through the break, put the broken DNA back together, and then we'll be able to release this passed segment here, what we call the transport segment. And then we'll be able to kind of reset the enzyme and release that again. That was pretty cool. Let's step back and just watch that part again here. So again, we have, of course, ATP is involved in this. We have a strand separation event here where we've actually made a double-stranded break in the DNA. We've cut both strands and we've pulled them apart. They're actually covalently bonded to the enzyme during this time. Pass the other segment through. We call this the gate segment, and this the transport segment. So the gate segment is now closed. The transport segment is passed through, and it's going to be released from the enzyme. So this is how we think this happens. We have some structural evidence to indicate this particular process and progression. But it's just really fascinating to see what's going on here. So this is kind of the summary of that again. You can kind of see that as that plays out. Now, why do we study these things? Why are we so interested in them? Well, it turns out that a number of anti-cancer drugs can disrupt the function of topoisomerases, like etoposide, which is very commonly used. Most of them tend to come in once the enzyme has cut the DNA and actually prevent it from continuing on through this progression. So you kind of get this stuck version of the enzyme where it's just kind of stuck on DNA. The DNA has been cut and it can't let it go. Now, that's great if you're trying to kill cancer cells. Obviously, that has bad side effects for healthy cells. And so that's one of the things that I'm interested in pursuing in my research is how do we make safer versions of these drugs that maybe are more selective in which topoisomerases they target and where they target them. Those are some of the things that I'm really interested in. So these are what I call the reversible molecular scissors of the cell. And I think they are, of course, fascinating. Come back next month, and we will talk more about topoisomerase 2, about its function, about its mechanism, about how it works. I can't wait to share more of that with you. But then I have to ask the question as we close here, what do we make of all of this? All these little machines at work in our cells, operating, functioning, doing the things that they do. Absolutely fascinating. And I think this is one of the really cool things that we get to see when we dig into and examine all that God has created and put here for us to discover. So thank you all for hanging in there. I hope I didn't put you to sleep too fast, but it has been a great time to get to share some of this with you. And I look forward to your questions as we kind of close out here. So, all right. So with that, I'm going to bring, so Andrew's going to come back up and join me here and would love to open it up to get some questions and comments from you all. So thank you.
All righty. Fantastic talk there.
Thanks, Andrew.
Joe, really appreciate how you highlighted the complexity that we see at the molecular level. And I think I'm just trying to read some of the comments here. There's a couple that have kind of come up. One of the comments was just about this idea. I think you said it at one point, shortly after looking at the ATPase pump, that this just happens. And the comment says something to the effect like that violates the laws of thermodynamics, because usually what happens is, I'm trying to summarize here for you, that things gradually stop happening. And, you and I both recognize that as the first and second laws of thermodynamics, in particular the second one with that one word, entropy. I think it's really just amazing to think about how the evolutionary story says, yeah, it just happened. Do they have any explanation for something like that?
Yeah, that's a good question. Of course, the classic, I guess, the classic irreducible complexity discussion, comes back to the bacterial flagellum. And the claim has been that, well, there are components of the flagellum that may participate in other, functions. And I asked Mike Behe about this one time, and he shared with me, he said, there are some parts of it that play roles in other complexes and certain strains of bacteria. He said, but that's kind of like saying that because you found the water pump, that you've got the whole engine. And he's like, the water pump's just one component. You still got a whole lot more to explain. And I would say the same here. You look at the ATP synthase, it's really sophisticated and has to be assembled at just the right time and place with just the right components. And that, it doesn't function without the gradient. Okay, so we need to create this proton gradient, which requires all the electron transport chain components. And of course, those don't transport electrons and pump protons unless there are electrons to be transported. So that requires, again, citric acid cycle. It requires glycolysis. And so you start to realize, okay, it's layer upon layer here that all have to be working and operating together to get that done. So yeah, that's a great point. Yeah, it doesn't just happen.
I was thinking yeah, and I think to date, it still begs the question. When we try to come up with an explanation of where this comes from, like you can go the one direction, like Mike Behe says, with you still have to explain the rest of the pump, but it's still there's nothing out there to date to say, okay, well, even if we said one of those was, okay, well, where did that come from? We still don't have any explanation. It's actually a science stopper when you stop to think about it. Now, a little bit lighter of a note, uh, where was the molecule of the month? How did you find that?
Yeah, so that's so the protein data bank. I think it's rcsb.org. I can't remember, but if you search on your favorite search engine, the protein data bank, there's a website that houses essentially all of the structures that are solved by scientists around the world. They get uploaded to this database, so you can go on there and freely search and look at these structures. And they pick out, yeah, there it is. They pick out structures to highlight in their work, and so they pick a molecule of the month. I assume they still do that every month, but they pick out a molecule to highlight and to talk about. And I just happened to, as I was looking for a way to illustrate the structure of the pyruvate dehydrogenase, turned out it happened to be a molecule of the month, and they had some pretty cool images. So I thought I'd share that. So we'll give a shout out to the protein data bank. So yeah, looks like another question has come in.
Yeah, I love the pictures that they share. They're just stunning. I have another question here from Terry. He's asking, how many amino acids are there in Topo 2 and the approximate molecular weight?
Yeah, great question. All right, so topoisomerase 2, let's go with humans, and particularly with what we call topoisomerase 2 alpha. So you have to get really specific because there's so many different versions. The one in yeast is different size than ours and so forth. But we're talking about a protein that's about 1,500 amino acids long, so 1,500 amino acids. It's actually specifically 1,531 is Topo 2 alpha. And that's in the order of 160 to 170 kilodaltons in terms of weight, 160 to 170,000 daltons. So it's a pretty big structure in terms of enzymes. A lot of times we talk about proteins that are maybe 300, 400 amino acids, kind of your average size protein. So this one's pretty large by comparison, and it's a dimer. As we'll talk more about next month, it's a homodimer, so it's 2 copies of that. So really, Terry, it's 1,300 amino acids, which is more on the order of over 300,000 daltons in size. So fairly big structure. And I know Terry in particular knows mass spectrometry. It gets really hard to study structures that large using certain mass spec methods. All right, looks like another question's come up.
All right, yeah, good deal. Here, I've got a question from Fred here. He is asking, can you talk more about the sensing systems that start the protein creation process? How does the cell know it needs to create a protein?
Oh, wow, Fred. That's a great question. How long do we have? So, we think about it, and I know.
Yeah, right.
Yeah. Andrew understands this too. You talk about the complexity of this. There's a lot going on, but let's just say for a moment. Let's go back to our sugar example there, and let's say our body has just we've just eaten something, and so all the nutrition from that is getting into our bloodstream and is being taken into cells, and so we've got insulin signaling. One of the ways that our cells respond to insulin signaling is by sending a signal all the way into the nucleus that says it's time to make these particular proteins, or really transcribe these genes so that they can be made into proteins for metabolizing the glucose. So you get activation of certain genes that are responsive and are needed for the metabolism of sugar. So breaking it down through glycolysis requires, like I said, 10 different enzymes. So there has to be a signal that goes to the nucleus to say, hey, it's time to make more of this. And so, there's kind of a mechanism there that tells the body, hey, we need this. It's time to make more of this. Cholesterol is another interesting example. There's a really, and we don't have time to go into all of it, but there's a really elegant mechanism in our cells for detecting the level of cholesterol. And when it drops below a certain level, there's a protein that gets transported from one compartment of our cells to another compartment. When it's in that second compartment, we call it the Golgi, it actually gets cleaved. And then once it gets cut into a couple pieces, one of those pieces can move to the nucleus and cause expression of the genes needed to cause the production of cholesterol. So it only moves from the ER to the Golgi when cholesterol levels drop below a certain threshold. And that's monitored specifically by the flexibility of the membrane, which is really bizarre to think about. So that's a very elegant thing. But Fred, yeah, we could talk all night about that, but there's some really amazing mechanisms there.
Yeah, I think about it just at the level of the E. coli that I've studied, where some of those sensing mechanisms, I talked with some people, and they say by the time you draw it up out of your micropipette tip, the signal has already sent and it's spiked, and you might actually get the wrong measurement if you don't treat it immediately, because it can happen in between 30 and 120 seconds. So this stuff is just rapid. I mean, how fast that happens, and it's required for life to occur. It's That's not right. Like, we can get by. Like, no, I mean, it's an either or kind of a deal. It is a matter of life and death.
Yeah, so, it is fascinating.
I got another question here from.
Yeah, let me say real quick before we go to this next question, Andrew. You mentioned, the speed of some of this. I mentioned replication earlier. I didn't say anything about how fast it is. So in eukaryotes, we typically think of it as being on the order of maybe 50 nucleotides per second are copied. And in prokaryotic systems, it can be on the order of 500 to 1,000 nucleotides per second. So, some of you who went through keyboarding and you were really proud of your, 120 words a minute or something like that, right? We're talking about 500 nucleotides per second, right? That's a lot of letters, all really, really fast, which is just fascinating. So yeah, go ahead on to the next question.
To keep going with that, that'd be like taking an entire encyclopedia volume set and typing it all out verbatim without any errors, getting it done in just minutes. I mean, it's
really
that's mind-boggling. Yeah. To think it just happened, like you said earlier, right. Like that just happened.
Yeah.
So John and Jenny Wise here have a question. Yeah, they're wondering, what scale does the mitotic spindle operate? Is it level the whole cell or the interior to the nucleus, or can you elaborate? I mean, mitosis is a kind of complex process, isn't it?
Yeah, that's right. So what we're talking about here, and if I'm understanding your question right, we're talking about cell division. And so we're talking about how does this kind of parent cell divide into what we call two daughter cells. And so at the point of the mitotic spindle, the nucleus has actually started to disintegrate, and we actually see it really disappear. Okay, so it no longer encapsulates the chromosomes, and then that spindle is able to form, and the chromosomes end up lined up in what we consider to be the center of the cell, where these microtubules extend out from the ends, the poles, in toward those chromosomes and attached to either side. And there's actually this dynamic tug and pull that kind of goes back and forth here, so that each one of those chromosomes all gets connected to opposite spindles, so that the sister chromatids are pulled apart. So one of the things that happens in that process, let me back up for a second. When our DNA gets copied during replication, we end up with two copies of each chromosome. and one needs to go to one and the other needs to go to the other daughter cell. So those have to be separated or segregated appropriately. So that's where the microtubules come in from the mitotic spindle, grab onto either chromosome, and then pull them apart during the process of mitosis. So that's going to be on the scale of the whole cell. And the nucleus at this point is really going to be, really has disappeared, because it goes essentially out of the picture until those chromosomes have been segregated into daughter cells. The daughter cells have pinched apart in cytokinesis, and then the nucleus can reform around those chromosomes that have been separated. So yeah, it's a whole cell scale. It's a great question.
Now, if I could, I'd like to interject and just to clarify just where your upcoming talk is going to fit in with that very question. Where does a topoisomerase fit in that whole process of mitosis? Is it when those get pulled apart, or is it before? Is it after? How does a topoisomerase fit into that picture?
Yeah, great. That's a great question. So topoisomerases are going to be needed at really multiple points throughout the whole cell cycle. So I don't have a diagram here. I'll try to remember to bring that in on the next talk, to bring a whole cell cycle so that we can kind of think about the stages that a cell goes through. But replication is going to be a major point where topoisomerases are needed. So as DNA gets replicated, there is this supercoiling that's going to be happening as a result of pulling the strands apart. And so we need topoisomerases during replication. We also see during replication that as those sister chromatids are copied, the sister chromosomes actually can wind around each other behind the replication fork, causing what we call catenation, or interlinking between the chromosomes. So if we don't have topoisomerase 2, those links do not get undone, and therefore the sister chromatids never get separated. They're stuck together. And so during replication, as you're alleviating the supercoiling in the head of the fork, you also have operations going on behind the replication fork to keep those interlinkings, those catenanes, or basically resolving them. And most of them we think are resolved by the end of replication. When you go into mitosis, there is still replication, or there is still topoisomerases involved there, because they are helping to compact the chromosomes into that final structure. And then they're also present during mitosis, so that there's no interlinks left between the sister chromatids that haven't been undone. So the topoisomerases are there too. So that's the role in a couple different points of the cell cycle. But you also have, before replication and between replication and mitosis, these two other phases. And during those phases, topoisomerases are needed to basically alleviate tension in the DNA so that you can get transcription of various genes. And so transcription doesn't occur. In other words, making RNA from the DNA doesn't occur unless topoisomerases are there to also alleviate that tension. So that's a good question. All right.
Yeah, when I think about topoisomerases, I typically think about from that diagram you had of the A going to B, going to C, and then back around again to A. Mm-hmm. And, I think it might be just worth putting out there that when we talk about what's required for a minimal cell, it's not just 3 parts. Like, it includes topoisomerase. And that topoisomerase has a very key role. And if that part, and it's not able to separate those strands of DNA, you don't have 2 cells. That's kind of, one of the beauties behind developing new drug targets to deal with this. We can develop new cures coming at it with a design approach,
Yeah, that's absolutely right.
So I have another question here. I don't know if you...
Yeah. Go ahead.
Okay. Sorry. I guess there's just a little bit of a delay. Apologize for that. Appreciate everyone's patience watching this. The question coming from Sandy asking, considering how convincing this is for intelligent design, the evidence that you presented here, which is overwhelming. Sandy asks, do you think intelligent design will be taught in schools again? What do you think, Joe?
That's a good question, Sandy. This is a very divisive topic, obviously, at large. While many of us are quite convinced by it, there are other people who aren't, and they have a very different perspective than we do. And so while I would like to think that that might be a possibility, we also have to be careful, because we may be biting off more than we can chew if we wish for that to be the case, because you could have some teachers who are opposed to it and who teach a very negative version of it in the school system. So that could happen as well. And so we have to be prayerful and careful about that. While I'd love to see that, I also am cautious about that, because perhaps the people teaching it might not want to represent it in the greatest of lights. And so that's something to consider as well. Now, should we give the freedom to do that? I think that at some point we have to consider that the science is enough, that it should, I think, demand that we're allowed to offer some alternatives to the current reigning paradigm out there. So that's just something we have to kind of think carefully about and prayerfully about. And we need to be, I don't think so much focused on pushing that particular approach, as maybe we need to be more diligent within our families and in our communities, making sure those around us are aware of this evidence and are considering it. So, yeah, it's great question, Sandy. Thank you.
Yeah, agreed. I think it's always it can be a very touchy subject if you start trying to think about the details to accomplish something like that. I always, I also have a hesitation that, I think a lot of times we want to present stuff like intelligent design, and it's a great argument, don't hear me wrong, but I think we have to remind ourselves, Paul wrote that these are invisible things are clearly seen, and then the author of the book of Hebrews reminds us that without faith, it's impossible to please God, and so that we, as Logos Research Associates, we want to make sure that we point people to Jesus. We don't want to just win the argument about design and leave people, just having won an argument. We want to point them to the Creator, Redeemer, Jesus Christ. So,
yeah, absolutely.
Yeah, I really enjoyed listening to you tonight, Joe, and getting a chance to interact with you. I'm not sure if anyone else has any additional comments or questions. I do want to talk up. I've known Joe for years, and I'm sure I've heard parts of the talk he's planning to give coming up here. And I can tell you, the levels of complexity that he has just talked about, if that blew your mind, just hold on and brace yourselves, because it says relax and unwind, but the truth is that the level of complexity is astounding, but it's not something that should intimidate us. Psalm 111 verse 2 is one of my favorite verses. It was etched in stone over the Cavendish Laboratory in Oxford. It says, "Great are the works of the Lord, sought out by all who delight therein." And if it's overwhelming, that's okay, because you know what? God's even bigger than all of this, and he pays attention even to the minutest of details. And so you can explore those depths to such great levels. And in the least, you can walk away with a better appreciation for just, wow, there's all that detail. And he gave us the tools to explore it. And some minds, just like Dr. Deweese, to even help us with developing new medicines are absolutely amazing. And so if you want to just be encouraged by that, I'd encourage you to come back again and listen to his talk about relax and unwind with reversible molecular scissors, DNA topoisomerases. And it will be a great opportunity for you to build your faith and invite a friend, maybe somebody who does not know the Lord Jesus or maybe has questions. I think ultimately, all of us, we don't have all the answers, but we know who does. And if every time we get together, we can just get a little closer to him, then we will have been successful. So I want to thank you again, Dr. Deweese. I have not seen any other questions coming in. I saw John and Jenny Wise comment how appropriately the infinite mind of the Creator will be our eternal playground. And I love the next comment coming in here. Science is more like, how does God do that? Reminds me of Johannes Kepler, thinking God's thoughts after him, and how we've advanced as a society built on a biblical worldview that promotes science and scientific thinking. So as a reminder, we want you to encourage you to go back to the Logos Research website. On the website, you can learn about the various research efforts, among those like Dr. Deweese and other associates and ambassadors, find out information about their publications. Please take a few moments, browse the website, consider supporting one or more of the exciting creation research projects like Dr. Deweese or others that are underway through Logos Research Associates. While you're on the website, please be sure you have subscribed to the Logos email list so you can receive notifications about talks like these and other upcoming events. When you subscribe, you'll also receive the Creation Evolution Headlines newsletter. We want to let you know that a recording of this video will be made available on our website so you can share it, and we encourage you to, with others who may not be able to join us live tonight. And again, just to remind you, our next talk will be by our very own Dr. Deweese again on Wednesday, June 12th at 9 p.m. Eastern. His talk title, like I said, Relax and Unwind with the Reversible Scissors, DNA topoisomerases. I'm now going to go ahead and close us in a short prayer. If you don't mind joining me, let's pray. Great God in heaven, how amazing and marvelous are your works. David wrote, fearfully and wonderfully made, and that my soul knows right well. And we affirm that. Taking a look at the reigns of DNA and the topoisomerases and how you left no details of this complexity unkept or untidy, and you took care of every detail. And so we praise you for that. God, if there's anyone here that does not know you, we ask that they would reach out and continue a conversation, and may they come to a saving faith in your Son, Jesus. Thank you for this time tonight. And we ask you bless Dr. Deweese with his research efforts and Logos Research Associates. We praise you in advance of it all, asking it in Jesus' name. Amen. So thank you all so very much for joining us, and we look forward to seeing you again in the next broadcast. Bye-bye.
Thanks.